Okay, so my article was over the efficacy of a Bruton's tyrosine kinase inhibitor, the PRN 473, in the treatment of canine pemphigus foliaceus. So, first of all, let me give you guys a little bit of a background for the experiment. Canine pemphigus foliaceus, which is abbreviated CPF, which is what I'm probably going to use through most of this. It is the most common autoimmune disease, skin disease in dogs and cats, and it normally presents itself in one of two forms. First, a pustular disease that causes ulcers and blisters, and second is a generalized dermatitis type, which is the normal dermatitis like scales, itching, redness, everything that follows. Um, these types of lesions normally begin to form along the bridges of their snouts, along the eyes, and around the outer parts of their ears. But in severe cases, it can spread along the whole trunk of the body, around the entire groin. And as you can see in the picture, it can engulf the entire foot along the claw bed, between their toes, and including the foot pads. CPF is caused by a type 2 immune response where the dog's body produces antibodies that attack specific transmembrane proteins, the DSC1. These are adhesive proteins that form the cell-to-cell junctions in the upper epidermal layers of the skin. And once these cells are damaged, it forms pustules on the skin's surface. And histology results have shown that these pustules are neutrophilic pustules. So they accumulate neutrophils in these pustules. The Bruton's tyrosine kinase, which is BTK, is a 659 amino acid protein that is used in human health to support humoral immunity. This enzyme is important in B cell signaling, but it also aids in the migration, development, and function of B cells. This enzyme is important because it was found that there is a 99% similarity in the BTK protein sequence between rodents, non-humans, or non-human primates, humans, and dogs, which makes this a great enzyme for them to test for immune responses between, in this case, humans and dogs. This study focused on BTK because autoreactive B cells are more dependent on this enzyme for survival than normal B cells. And they found that this out through other testing, and they found that when the BTK is absent, there's a loss of autoantibody within the body's total antibody count. And since the pemphigus foliaceus is an autoimmune disease, the scientists wanted to focus on this protein for their study. BTK inhibitor drugs inhibit the BTK in all non-T cell white blood cells, which, as we all know by now, include B cells, neutrophils, mast cells. These drugs decrease the downstream signaling from B cell receptors and surface IgG receptors on the white blood cells. The inhibitor drugs also reduce the ability of B cells to mature, which in turn will interfere with their ability to proliferate, differentiate, and survive at that point. So this has made the BTK inhibitors important in treating certain forms of cancers in humans as well as in dogs. So for this study, the scientists decided to use the PRN 473, which is an investigational BTK inhibitor that is poorly absorbed in humans, but is well absorbed in dogs. Since BTK is crucial for B cell signaling as well as autoantibody production and survival, the thought was that the PRN 473 enzyme could possibly be effective in regulating and stopping the proliferation and survival of the autoantibodies that cause the CPF. The main objective that they were aiming for was to determine the efficacy and safety of the PRN 473 in dogs with naturally occurring CPF. And for their hypothesis, they considered that by giving the patients this PRN 473, the B cell signaling would decrease, the autoantibody production would be impacted, and less anti-DSC1 would be produced. And just to remind you that the DSC1 is the adhesive protein that holds the skin together. In this experiment, they used 9 dogs that were diagnosed with naturally occurring CPF. To be considered, they had to have shown little to no improvement after being treated with antibiotics specifically for this disease or appropriate for this disease, as well as not being given immunosuppressive medications of any type. At the start and throughout the study, blood samples were taken from the jugular vein, and sampling times ran from before being administered any medications, which would be at 0. And they were taken after administration at 4 and 24 hours, as well as at 2, 4, 8, 12, 16, and 20 weeks. The peripheral blood mononuclear cells, which are the PBMCs, were collected, centrifuged, and washed. And just to clarify, the peripheral blood is any fluid that travels through your heart and arteries and veins, along those lines. And mononuclear cells are blood cells that have a round single nucleus. Now, these include monocytes and lymphocytes such as T cells, B cells, and NK cells, but neutrophils, basophils, and other granulocytes were not included since they have multi-lobed nuclei. And after the PBMCs were washed, an aliquot was removed and labeled with an anti-CD21 antibody, followed by a donkey anti-mouse IgPE, which is a bright red fluorescent protein, in order to help detect the B cells. And these labeled cells were read using flow cytometry in order to determine the percentage of B cells within the whole lymphocyte population. The remaining PBMCs were tested using a competition assay, which used a fluorescently labeled BTK probe in order to find the percentage of BTK enzyme that was bound by that PRN inhibitor. These probed enzymes were then run on electrophoresis gel and calculated against the total BTK count. All of these tests were performed for each blood sample that was taken. So for each dog, for every time point. The IgG titers for the adhesion protein, the DSC1, was calculated by performing multiple dilutions. If the proteins were present in that dilution, they would present with a color change for a positive result. The higher the dilution in which these proteins were present showed that there was still sufficient adhesion between the cells and that the BTK had little to no effect. If the dilutions went down, then there were less adhesion proteins available to hold the cells together. So these IgG titers were performed before and at the end of the treatment period for each dog. So they went through the entire treatment before being tested again. The PRN 473 inhibitor was administered through an oral tablet that was given daily with food. The dosage was approximately 15 milligrams per kilogram of body weight. If a dog showed no change in the lesions on its body after about 2 to 4 weeks, or if the BTK percent analysis showed below the results below what they were wanting, then their dose was doubled. If the dog showed positive results after 12 weeks, the goal at that point was then to reduce the dose being administered to every other day for the remainder of the treatment period. The dogs were monitored regularly to ensure safety by testing complete blood counts and levels, running biochemistry panels to ensure proper minerals, vitamin levels, and all the basics were maintained, and evaluating their urine for a proper concentration levels appearance. Each dog was evaluated at 2, 4, 8, 12, 16, and 20 weeks and rated on their response to the treatment by using a human pemphigus disease activity index, which is the CPDAI, that had been modified for this canine use. The results were measured by one of 3 different categories, where good was greater than 90% of the lesions were resolved by the drug alone. FAIR was greater than or equal to 50% of the lesions were resolved, but corticosteroids were also required in order for the dogs to maintain comfort levels. And poor was when there was no improvement in the lesions and there were additional need for some of the traditional medications for that disease in order to gain relief. The evaluations using this index were performed by two different veterinarians who were blinded by each other's scoring, and the results were then averaged. A Wilcox rank sum test was used to determine the correlation between response to treatment, the percentage of BTK inhibitor noted at the 4 and 24-hour mark, as well as titers to the anti-DSC1 protein. And these results were calculated by software. And a p-value of less than 0.05 was calculated as being significant. At the conclusion of the 20-week treatment, there were mixed results from the dogs in the trial. 5 dogs exhibited good results, with 3 dogs showing near complete remission of the lesions by the end of the treatment. Dog 5 was removed at 12 weeks due to development of immune-mediated polyarthropathy, which is an arthritis or joint pain affecting 5 or more joints at the same time. Dog 1 also showed signs of this, but was able to continue with the study. Dog 8 was removed after 4 weeks due to the recurrence of a previously removed mast cell tumor with additional spread, but this dog was also showing good results when it had to be removed. 2 dogs exhibited fair results, but were removed from the trial for not responding quickly enough to the treatment. Dog 7 was removed at 8 weeks and 9 at 16 weeks, and they both additionally required some type of intervention with other immunosuppressive doses of like a prednisone, what's it called? Prednisolone, I think it was, to help alleviate the pain and inflammation. And 2 dogs were removed from the study due to no improvement after being administered treatment. Dog 2 was removed at 8 weeks and dog 6 at 16 weeks. Both of these dogs also required the prednisolone treatment in order to help gain some type of relief. Dogs 1 and 3 were the only 2 patients that had good results that were able to have their doses reduced to every other day, which was one of their goals that they were trying to reach while administering the medication. Dog 1 continued to remain symptom-free for 6 months after the treatment period before relapsing, and then was started on that PRN inhibitor drug again. For the anti-DSC1 titers that were obtained, there was a decrease in dogs 1, 4, and 5, which all showed good clinical responses. Dogs 6 and 7 showed an increase in these titers, which all showed, fair to poor clinical responses. And there were no detectable titers in dogs 2, 3, and 9. And dog 8 was removed for the mast cell tumors before this test was performed to see what the titers were. So the role of the PRN473 inhibitor was presumably to interfere with multiple inflammatory pathways, one being neutrophil migration, which CPF is known to show an infiltration of the neutrophils, which causes those pustules. Another is, which I already spoke of, to slow the function and proliferation of B cells. And lastly, it was assumed to decrease the production of autoantibodies that target transmembrane proteins, such as the DSC1. After the completion of this experiment, no real correlation was shown to exist between the good response and the BTK inhibitor percentage. A possible reason for this could have been the metabolism of the inhibitor between different dogs, or maybe even different breeds. The cause of the immune-mediated, the arthritis pretty much, was unknown as to whether it stemmed from this drug or if it was simply due to old age. Because dog 1 was 11 and a half years old and dog 5 was 12 and a half years old. So it's very possible that these dogs had the arthritis before this study. The recurrence of the mast cell tumor was unlikely due to this drug, because other studies have shown that the BTK inhibitor molecules actually have anti-tumor effects on canine mast cells and other types of lymphomas. So that was kind of concluded as not being related to this drug. So the study showed that the Bruton's tyrosine kinase inhibitor PRN473 was at least moderately successful in 7 of the 9 dogs that were given the treatment. This would be a good candidate for future testing involving larger quantities of test subjects, using a variety of different breeds for comparison of the BTK inhibitor metabolism and responses, as well as having a proper control group, because this test did not have a control group. If the drug alone was not sufficient in treating canine pemphigus foliaceus, then it could definitely be further tested to become a potential supplemental drug to aid in the treatment of this disease. And here are my references.
Great. So I guess we'll hold off on your questions, and you might have sent these to me already. So any questions for Shelley in her presentation? Yeah, I was a little bit, I'm glad you mentioned the number, because I was getting nervous with just 9 animals. You know, how much of that was due to chance, for example?
Exactly. And that's what it seems like, because I was going to post some of the charts and stuff that were on here, but they were so tiny, you couldn't really read them. Mm-hmm. But they had different breeds that they seem to think respond a little better, but with this number, they can't really get the correct answers they're looking for in this type of study unless they do a larger, more spread out to show different types of breeds, different ages, different everything, different life stages.
So were you convinced that this was a useful drug based on what they presented?
I think it's got potential. And that's why I had said in like my kind of conclusion to this, that if the drug alone isn't sufficient for treatment, then I definitely think it could possibly be used supplementally to help, along with the traditional drugs that they normally use for like different corticosteroids and stuff. Mm-hmm. Because if it does inhibit those autoantibodies, I definitely think they do need to do a lot more studies on it. They need to do a bigger study to show that it does in fact work the way they think it does. But I mean, this little study looked promising, but it definitely needs to be sprawled out more.
Any questions for Shelley? I have one more for her. You can also type them on the chat box. I can actually see it today. So one other question I had with regard to, did you say that the anti-DSC1 titers correlated with their response clinically? So if they had, fewer titers against DSC, did it correspond with better clinical outcome?
They had, in the good responses, which you would assume has the anti-DSC, so you would have a lower number that would be good, because you don't want that to associate with the to break down that cell-to-cell bond. And it did, I do believe that it said that the anti-DSC titers for the dogs with the good clinical responses was decreased in those dogs. So if I'm thinking along the right lines, I think that pretty much pertains to what we're looking at, right? Because you want the anti-DSCs to be lower. So if they had the good response, yeah, and I think for the most part they did. There were a couple that they could not get any titers on, And I think there was one of the dogs with the good response that they didn't get any DSC titers on.
Yeah,
They
so I don't even know what that means. Like, so does that mean that they were completely, did they do pre-titers and post-titers after treatment?
that, I kept looking for different things and they didn't really explain. I tried to find other things about this in other um experiments and stuff, but I really couldn't find a whole lot. So I don't know. There was a lot of questions in this
Mm-hmm.
that I went back through and kept reading over stuff and trying to find little things of why they did this or why they got this answer, and they didn't really explain
Mm-hmm.
a lot of those.
It sounds a little bit weak. I mean, it sounds like the
Yeah.
paper just maybe a little bit on the weak side, especially with, the numbers of animals. I think they could have probably enrolled a few more than 9, but
Yeah, I definitely think, I mean, from reading about the inhibitor drugs, I definitely think it's something that could be researched more to
mhm,
see if it could be useful in this. But if
mhm,
they want to use this study like this, they definitely need to expand it out.
right. Great. All right. Well, I think we better go on to the next presenter. Thank you, Shelley. If you wouldn't mind stop sharing, that would be great. So, and also autoimmunity and those types of diseases are going to be covered in the next couple of weeks with Dr. Imwangi. So I'm glad you're kind of preempting that. So, all right. So, Rakshith, I think you are next. Do you want to share your screen or your presentation?
Yes, Dr. Adams, I do.
So you're going to be talking. Oh yeah. Oh, good. African swine fever. Great. This is an area of this is where you're, the lab that you're in?
Yes, yes. Actually, this paper is from our lab only. So this is like the first step for our lab. To start,
Okay, yeah.
we have to read this paper and understand this so we can start.
Great. Great. Go ahead and you can start anytime.
Good afternoon to all. So today I'm going to present this paper titled Induction of Robust Immune Responses in Swine by using a cocktail of adenovirus vector African swine fever virus antigens. So basically, we have developed a vector-based vaccine for African swine fever by using the specific antigens, so which I'm going to tell in the next slides. So before going to presentation content, so first I will give the introduction about the African swine fever disease, African swine fever virus, vector-based vaccine, and different platforms in the vector-based vaccines which are currently being used. And then I will talk about the methods and results which are discussed in the paper, and then I'll summarize and then I'll discuss the strengths and weaknesses and the future studies and questions. African swine fever, it is a highly contagious fatal hemorrhagic disease in swine, which causes the morbidity and mortality of 100%, and the recovered swine will become carriers, and there is no treatment or vaccine available till now. The only preventive or the control measures are quarantining the swines and removal of the infected and the animals which are in contact with the infected pigs. So these are the typical lesions which are seen in the African swine fever disease, that is hemorrhagic lesions on the skin. So this is very similar to the classical swine fever also, which are very similar. So African swine fever virus, this virus belongs to Asparviridae family, genus Asparvirus. Size of this virus is 170 to 190 kb. It is having non-segmented genome and it is an enveloped virus. So in the structure, you can see the outer envelope, um, capsid protein, inner envelope. So this is the genome which is covered by the nucleoid and the matrix, and then the inner envelope. So these are the different proteins which we'll use for the development of the vaccine. So these are coded by the specific genes. So we'll take them and put in the virus backbone, and then we will produce the protein which are specific, which will elicit the immune response for this virus. So the transmission of this virus is by the ornithodoros tick in the wild conditions to the wild suites, specifically the warthogs. And these warthogs will not get any infection, but they will act as carrier and then they will infect the domestic pigs. So there is no any treatment or vaccine. So before going to vector-based vaccine, I will tell about which are the different type of vaccine which are available, which we discussed in our Monday class. So there are live attenuated vaccines, inactivated vaccines, subunit vaccines, recombinant vaccines, and toxoid vaccines, and etc. And and in the recombinant vaccine, we use a specific epitope which will elicit a strong immune response. As I said, so we'll code the specific antigen or the epitopes which are present in the virus to elicit the immune response. Vector-based vaccines. So in this paper, we have used the vector-based vaccines. So these vaccines uses the attenuated or the inactivated viruses. The viral vectors will enter into the cell and uses the host molecular system and produce the desired proteins to elicit the immune response. So they naturally mimic the natural infection. So the virus will enter into the body and produce the immune response, but which is not lethal. And the advantages about these vector-based vaccines are they are easy to make and they are cheaper to make. Example, the adenoviral vector can be grown on cells and used for different vaccines. And one vector is the same for all vaccines. Like if we prepare a one best vector, one backbone, so we can insert any gene and then we can develop the vaccines. And these vector-based vaccines triggers a robust, innate, and adaptive immune responses. So currently used vector-based vaccine platforms are the adenovirus, which are being used in HIV and influenza vaccines. Vaccinia virus vector is used in smallpox. Lentivirus vector is used in HIV. Cytomegalo in SIV. Sandivirus vector, Sandivirus vector is used in HIV vaccine. Retrovirus vector is used in malignant glioma, which is a cancer. So coming to the methods and the results which have been discussed in this paper. So there are this paper is a little big, but I will try to finish within the time. So first they have developed a plasmid which have our pro which is having the gene of interest or the gene which is producing a specific protein, and this plasmid will be transformed into a PAD vector or the adenovirus vector, and this adenovirus vector is evaluated in laboratory condition in vitro by using the cell culture, and by these results this has been translated into uh, in vivo by giving the vaccine to the swines, and the swine serum samples and blood samples have been collected to evaluate the antibody response and the T cell response by conducting the ELISA, IFA, and CTLSA. So first generation of the plasmid constructs encoding the African swine fever antigens. So we used pcDNA. It is used to generate the DNA plasmid constructs. So this is a pcDNA and this is the gene of our interest, which will produce P32 protein or P54 or PP62 or P72. So P32 is a phosphoroprotein. It will help in the viral entry in the natural infection. P54 is also helping in the virus entry. PP62 is used in the viral core development, and P70 is the major capsid protein. So these are the proteins which have been thought that like these might be the protective antigen which will produce the robust immune response or the protective immune response. So we further and then these plasmids were and these genes were tagged with flag tag and HA tag for the further immunocytometric analysis and all. And this pcDNA is used to develop the adenovirus vector-based vaccine by transformation method. So for transformation, we are used the PAD is used to generate the recombinant adenoviruses. These PAD constructs were validated by immunocytometric analysis, which we'll discuss in the next slide. And these positive constructs, which have been positive in the immunocytometric analysis, were scaled up to generate more and more virus for the immunization. immunocytometric analysis, so evaluation of the protein. After developing the virus, so the protein expression by the plasmid constructs, which we done in the first step, and the recombinant viruses, which we did in the second step, were evaluated by immunocytometric analysis. So this is a schematic diagram which I made. So the transfected by the plasmid constructs and infected with the recombinant adenoviruses. And so the 293A HEK cells were transfected or infected, and the primary antibody were used. The one used was the anti-flag AP, and the other was the anti-ASF. These two are the different procedures. For mouse anti-flag AP, there is no need of secondary antibody because it's already having the alkaline phosphatase here. So by adding the fast red, it will give the color like this. So this will show the presence of our protein. So these are the analysis, like if our protein is being expressed in the cells or not, like that. So the second confirmation is by probing with the anti-ASF serum, and then probing this with goat anti-swine AP, which is having the alkaline phosphatase. And these two, if if our protein is present in these cells, or the proteins have been produced in the cells, so we will find the color. So the results are like this. So this first row is showing the transfection, and these two are showing the infection. So the first one is showing the anti-flag antibody. So P32, P54, P62, P7, all the four proteins were produced after the transfection of the plasmid, and after the infection of the PAD virus with our gene of interest. Also, the anti-flag antibody was showing the positive signal, and also anti-ASF, which we saw, anti-ASF and anti-flag. So we have done the double check. So first is anti-flag and second is anti-ASF. So both are showing the positive color, but the negatives are showing nothing. Means so there is no virus or the protein producing the negative controls. After confirming with the in vitro analysis, so the immunization of the swine is performed. So four groups were were done, and each group were having five piglets. So three groups, T1, T2, T3, were injected with a cocktail of recombinant adenoviruses, and one sham, or the last P4 group, was infected with adenovirus elusive virus, so which is not having our protein of interest. So the uh, route of administration was intramuscular route, and the first group received 4 into 10 to the power of 10 dosage plus ENABL adjuvant, and T2 received higher dosage 4 into 10 to the power of 11 with ENABL adjuvant, and T3 group received 4 into 10 to the power of 11 dosage with Zoetis adjuvant, and T4 received only Idluc virus. So the booster dose was given after 14 weeks of first dose, and all the piglets were sacrificed after 8 weeks post group. So T1 is a lower dose group, T2 is a higher dose group, T3 is having Zoetis adjuvant with a different adjuvant, and T4 is a control. So the antibody responses were evaluated by ELISA. So ELISA is very, so we have been already learned. So the ELISA plate, so we coated with a specific antigen, P32 or P54, P62 or P72. And then the serum sample, which is collected from the immunized swine, were collected, and then like we added into the ELISA plate. And then anti-swine IgG were was added, which is a secondary antibody which is conjugated with a peroxidase. And upon adding of the substrate, so it gave color. So this is a mechanism of ELISA. So the results were, so for P32, both IgM and IgG responses. So for all the four proteins, which are in the next slide, so P32, P54, and P62, P72, both IgM and IgG responses were evaluated. The lighter bar is showing the IgM response, and the darker bar is showing the IgG response. So by seeing the results, we can tell that there was production of robust African swine fever versus antigen-specific antibodies, and 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 by seeing, we can see within 1 week there was the presence of more of IgG antibodies than the IgM antibodies, because the second bar is topping the first bar, which is the IgM bar. So this result showed that the class switching of the antibodies from IgM to IgG in the first week itself, in T1, T2, you can see. And to be specific, the P72 protein, the pigs showed in T3 group showed highest responses. And also we did the biweekly monitoring of antigen specific IgG response. So each bar represents a 2-week interval. So the first bar is the second week, the second bar is fourth week, like that 6, 8, 10. So the antibody responses were peaked at any. So this result showed that the antibody responses were peaked at any time between 2 to 8 week and gradually declined by 10th week for all the proteins. So we can see this gradual increase in the antibody responses, and by the 10th week there is gradual decrease in the antibody response. But the P72 specific antigen responses were highest for T3. You can see the tall bars, and it is slightly lower in the T2 animals, and the lowest in the T1 animals in case of P72 protein. So these both results will show that the ability of the vaccine cocktail to rapidly induce the IgM and IgG responses in a single dose. So these all results are from the prime dose only, means only the single dose. And post-boost IgG titers were also showing the same results. So they have not much spoken about it. And next, to evaluate the production of the ASF virus-specific antibodies, they conducted the indirect fluorescence antibody assay, or IFA assay. To give a brief information, the Teflon-coated slide wells. Uh, so first of all, they collected the macrophages from the pigs, and they infected the macrophages with the African swine fever virus. And these infected macrophages were coated on Teflon-coated slide wells, and then the swine serum was added to the wells, and fluorescein isothiocyanate conjugated goat anti-swine serum was added over that, which was having the fluorescein dye, which will give the fluorescence upon excitation. So the results, so the positive control serum all showed the results, and the vaccines also, means the vaccine groups were showed the fluorescence present. But the negative control animals didn't show. So this shows that this recombinant uh, vaccine induced the authentic African swine fever virus specific antibody responses. And to evaluate the T cell responses, chromium release assay was performed. So for performing the chromium release assay, we have to generate the effector cells, and we have to generate the target cells, and then upon mixing the effector cells and target cells, there will be release of the chromium dye. So by evaluating this, we can tell the action of the T cells on the infected cells. To generate the effector cells, the PBMC were isolated from the blood on 4 week 4 week post boost, and they were infected, or infected means they were primed with the recombinant adenoviruses at MOA of 1000 for stimulation of the T cells, for the first priming of the T cells. And then the target cells were produced by collecting the skin tissue, and then these means the fibroblast culturing was done with the sodium chromic oxide, and prepare the target cells. So the effector cells are the PBMCs, and the target cells are the fibroblast cell culture with the chromium. And then these both effector cells and the target cells were mixed at the ratio of 50 is to 1 and 25 is to 1 for different evaluation. And then the chromium release was evaluated with a micro beta counter. So the lytic, so when the effector cells will act on the target cells will die, and there will be lysis of the target cells, and the chromium will be released, and the release of the chromium will be measured to know the lytic activity of the PBMCs. So the lytic activity observed against the African swine fever virus antigen can be attributed to the African swine fever antigen specific T cells, and then and by looking at the results, the lytic capacity of uh, was similar in T1 and T3 groups, but it was not in the T2 group. So this was the result. To summarize all the results, the recombinant adenovirus African swine fever multi-antigen cocktail 2-dose formulation was immunogenic and safe when administered in prime boost regimen. The results showed the rapid post-prime antibody class switching, induction of the robust antibody responses which recognize the African swine fever infected cells, which we saw in the IFA, and also it showed the generation of the antigen specific fetal responses, which we saw in chromium release assay.
Rakshith, I think I'm going to get you to stop here and see if we have any questions from anyone, just due to time. Seems like a really interesting study and very convincing, and it really kind of really brings immunology up, because you're talking about a bunch of the immunoassays that we talked about, including chromium release, which is great, as well as the vaccines, the different types of vaccines. So I think that's pretty neat and looks like some exciting data. Does anyone have any questions for Rakshith? So one question I have is, have you actually put it in, have you challenged these animals with this cocktail?
Yeah, actually the further study was the same. So the result was they were actually showing the immune responses, but the piglets died. Means the vaccine cocktail is not protective. So these proteins are not protective.
Yeah,
So
that's the frustrating part, isn't it? Trying to find the protein that's going to provide protection.
Yes.
That's so you still have there's more work to be done, it sounds like.
Yes, yes. And also we are finding the protective antigen now. Like we are
Yeah.
reading the papers and all. Yeah.
Yeah, that's challenging. So it should be, but this is a really great study. It seems very complete. Unfortunately, the challenge did not work, which is frustrating. All right, so I'm going to get you to stop sharing your slides so we can move on to the next presenter, which is going to be Vanessa, and you're going to be talking about COVID.
Yes.
Yeah, that's great. Thanks, Rakshith.
Thank you.
Great study.
Okay, let's see if I can do this too. Oops. Share. Oh. I'm having some technological issues.
So when you click share screen, what are your choices?
It's asking my system preference to allow Zoom to share my screen, so I need to remember my password real quick.
Should we go on to the next speaker?
I think maybe until I figure this out.
Yeah, until you get it figured out. Can you get it figured out after Emily presents?
I believe so.
Okay, let's do that. So Emily, if you don't mind stepping up.
Sorry.
I've never heard of this. Yeah, some extra security, but
No problem. Sounds good. Let me move some stuff off my bench real quick.
okay.
I had some stuff in my way. Alrighty. Let me remember how to share a screen. Can you see my slides?
Yes.
All right. Try to. There it goes.
You got it.
Okay. Yeah. Move you guys out of the way. Can you hear me all right?
Yes.
Okay. So the paper I am presenting on is unexpected role of CD8 T cells in accelerated clearance of Salmonella enterica, Cerevar typhimurium from H2 congenic mice. This is published in the American Society for Microbiology, Infection and Immunity in November of 2019. A little bit of introduction about Salmonella. It is a gram-negative rod bacteria. The CDC estimates for the US annually that there are about 1.3 million infections, 26,500 hospitalizations, and 420 deaths. It is the most common source is contaminated food or poultry. And it is currently ubiquitous in the US poultry industry, so you're going to find it in every complex. There are two types, non-typhoidal NTS salmonella, which is your gastro and enteric infections, and typhoidal salmonella, which cause typhoid and paratyphoid fever. Salmonella is separated into about 2,500 different serotypes, and these are determined on the somatic and flagellar surface proteins on the bacteria. So some major ones that we see in humans on the human side is Enteritidis, Typhimurium, Heidelberg, Infantis, Kentucky, and many more. So we're going to be focusing on the Typhimurium for this study. Salmonella pathogenesis goes kind of like this, where I'm going to see my little clicker here. You have Salmonella that's introduced to the intestinal tract orally, so it's floating around in the lumen of the intestines, and then it exploits the intestinal epithelial M cells to gain entry into those Peyer's patches, where they then begin to infect the dendritic cells and the macrophages. They then migrate into the mesenteric lymph node and blood via the lymphatic system. There are some circumstances where they can also infect via breaching the epithelial barrier and disrupting tight junctions, so getting between the cells as well. But once they are in, the infection is seated in the intestine, it rapidly spreads to systemic tissues where it can replicate in the liver, spleen, and bone marrow. And in this study, we'll be focusing on the liver and the spleen. So when there is a infection with Salmonella, there are multiple immune responses. And the immune responses are initiated rapidly after infection because infection can happen very quickly and it's affecting the Peyer's patches. The major mechanism of infection, of the immune response is activation of macrophages of T helper 1 cell secreted gamma interferon. And so CD4-TH1 cells play a large role in this. And it is the main lymphocyte in congenic mice. Mice lacking the CD4 T cells have delayed bacterial clearance and had higher bacterial burdens after a month of infection. So these do play a major role. Infection of susceptible mice with an attenuated strain of Typhimurium elicits a robust CD4 T cell response that contribute to bacterial clearance, and infection of resistant mice with a strong, with a virulent Salmonella typically elicits a strong antibody-mediated protection. There are also other protective mechanisms that may be important. These are ones we're going to look at. In this study, they hypothesized based on historical data that a stronger protective CD4 T cell response would develop in the C57 Black 6, which is a control mice that are expressing other major histocomplex haplotypes. So for this study specifically, they want to examine whether the H2 congenic mouse strains with enhanced resistance to Salmonella infection elicited a superior CD4 T cell dependent protective response. So the mice used in the study were the C57 Black 6 mice. They are commonly used inbred lab mouse that it has the most widely used genetic background for genetically modified mice. They also use H2 mice, which are mice expressing the various H2 MHC haplotypes. In this study, we look specifically at the H2B, H2D, H2K, and the H2U haplotypes. The H2 genes encode for ubiquitously expressed transplantation antigens that serve as recognition structures for cytotoxic cells. The H2K and H2D regions of the H2 gene complex are highly polymorphic and control cell surface structures involved in the H2 restriction of cytotoxic T lymphocytes. So congenic mice are inbred mice where part of the genome of one mouse, the donor mouse, is transferred to another. And this is usually done by back crossing to the receiver strain with specific selection for the desired gene. So we're looking at these mice here on the right-hand side where they have the majority of the receiver DNA, but there are some alleles that have been changed. And the alleles we're looking at are those H2B, D, K, and U. The salmonella used in this study was BRD 509 Arrow A strain of Salmonella enterica cerevar typhimurium. This is a metabolically attenuated strain, so it's not virulent. And the SL1344 ATCC strain of Salmonella typhimurium. This is a common non-attenuated reference strain. It is considered virulent. The methods and results for this study. They looked at multiple things. So the very first thing they wanted to look at was salmonella clearance and make sure that they were seeing the same results as previous studies. So specifically, they wanted to see whether the MHC congenic mice displayed a different rate of salmonella clearance. So they used mice possessing the variant H2 molecules at the class 1 and class 2 alleles, as well as congenic control strains. And these mice were all infected intravenously with 5 times 10 to the 5th colony-forming units of Salmonella typhimurium. The bacterial burdens of the mice were assessed over 28 days. So first off, we are looking at the salmonella clearance of the H2D and the H2B alleles. And so what you're looking at here is the samples from the spleen are on top, the liver is on the bottom, and the H2D is here on the left, and the H2B is on the right. The purple is the different H2 samples, and then the black is the control. And in these, we can see that there are no significant differences were observed in the rates of clearance. They're very close from the spleen or the liver of the control strains or from their MHC congenic counterparts. So therefore, allelic substitutions of B and D haplotypes at the MHC locus had no discernible effect on Salmonella typhimurium infection. However, when we look at the salmonella clearance of the H2K and the H2U haplotypes, we do see a lot of a difference. Same setup where the spleen is on top, liver bottom. The two in C are the H2Ks and D is the H2Us. And mice with the H2K and the H2U haplotypes cleared Salmonella typhimurium from the spleen and the liver at a higher rate than the control mice by 2 weeks post-infection. So you can see here, I'm looking at the spleen, that there's already a significant difference at 2 weeks, and you see that trend through all of the graphs. Then by 28 days of post-infection, there was a 100-fold difference between the H2 haplotypes and the control mice. And you can see that on every one of these. So therefore, the MHC alleles H2K and H2U do influence the ability of mice to resolve infection with Salmonella typhimurium. So next, I wanted to know whether the functional capacity of T-bet expressing Th1 cells, how that affected this infection. T-bet expression is necessary for resistance to Salmonella because it is a transcription factor that differentiates Th1 cells. In this study, mice were infected with Salmonella typhimurium, and then 2 weeks post-infection, they were stimulated with a lipopolysaccharide derived from purified E. coli in order to activate CD4 and CD8 T cells. They use flow cytometry to monitor the Th1 cells and the effector CD8 T cells. So here, B is showing the interferon gamma production here at the bottom in the CD44 T-bet CD4 and CD8. Sorry, kind of get a little switched up there. T cells. And then D here is showing that approximately 60% of the T-bet and CD4 T cells from the H2B mice here in this first column, they produce interferon gamma, which is indicative of a strong Th1 response to Salmonella typhimurium. So you can see that those that were given the lipopolysaccharides had a very much higher percent of interferon gamma production when compared to those that were not given it. And those are for the CD4, sorry. These are for the CD4 cells. And you can see there's about 60% here, 40 to 50, and 20 to 30. So you're looking, there's quite a increase in CD4 response. But when you look at the CD8 T cells, you're only looking in the 1 to 2% kind of range. So there's a much lower rate of or percent of interferon gamma production in the CD8 T cells. But I'm sorry. So as expected, though, the level of CD8 T cell production in all the mouse strains was considerably lower than that seen in the CD4 Th1 cells. But the H2B and H2U had similar frequencies of interferon producing CD8 T cells. There's about 1 to 2%. But the H2K had increased frequency of interferon producing gamma producing CD8 T cells, which correlates with a greater role of CD8 T cells mediated control of Salmonella typhimurium infection. And so these are looking at about 2 to 3%, which is twice of what the 2B and 2U were showing. So overall, this data suggests that greater protection against Salmonella infection in these MHC congenic mouse strains is likely to be independent of Th1 interferon gamma production. So there must be something else going on. Next, they wanted to directly test whether this accelerated clearance was dependent on CD4, CD8, or the combination of both CD4 and CD8 T cells. So from here on out, they're going to focus these studies on the H2K congenic mouse strains. So for this study, they infected the mice with Salmonella typhimurium, and then they had different groups that were treated with either anti-CD4 antibody, anti-CD8 antibody, anti-CD4, and anti-CD8 antibody, or an isotype control. The mice were treated with their allotted antibody or control over 3 to 4 days from day 7 to day 30 post-infection. And these results were determined by flow cytometry. So in this graph, we have in the blue, we have the spleen, and in the purple, we have the liver results. The H2B is on the left and H2K is on the right. The black bars are your isotype control, and then it goes CD4, CD8, and the combination of both CD4 and CD8. So with regards to H2B mice, the bacterial burden here was shown, the depletion of both CD4 and CD8 T cells in the H2B mice did not show much difference when compared to the effects of depleting just the CD4 T cells. So you can see here this dark blue, there was no significant difference between that and the lighter blue, and same with the purple down here in the liver. However, the depletion of CD8 T cells alone resulted in increased burdens in the liver. Thus, the CD8 T cells made a small contribution to primary clearance in the H2B control mice. When you look at the H2K mice, depletion of both the CD4 and the CD8 T cells resulted in significantly greater bacterial growth than CD4 alone. So this is showing the clearance. So you had higher, anyway. So in the spleens here, CD8 T cells provided more protection than the CD4 T cells. individual depletion of CD8 T cells had a stronger effect. In the liver, it appears to provide redundant protection since depleting either the cell types did not result in loss of protection. You can see when compared to this isotype control that there's not a significant difference. Oops. So therefore, CD8 T cells play an important role in the accelerated bacterial clearance observed in the H2K expressing MHC congenic mouse strains. So next they wanted to see if the functional markers of CD8 T cell activation and effector differentiation may explain the protective capacity. So that using flow cytometry, they looked at CD8 T cells with this KLRG1 is a cytotoxic marker, and the CD4 T cells with this PD1 it's an inhibitory marker for the flow cytometry. And they monitor this at day 14 post Salmonella typhimurium infection. So here what you're looking at is the spleen is here on the left and the liver, the liver on the right. The black bars are the H2B, which were used as the control in this situation, and the blue is the H2K. And so at 14 days post-infection, there was a significantly greater proportion of CD8 T cells in both the spleen and the liver of H2K mice. You can see how the blue bars compare to the black controls. There was also significantly fewer CD4 T cells in the spleen of the H2K congenic strain. So you can see how those are much depleted. So therefore, the increase in CD8 T cells and the decrease in the CD4 T cells suggests an overall enhanced pro-inflammatory state in the H2K mice, which was likely responsible for the accelerated clearance of Salmonella in this congenic strain. So finally, they wanted to see, well, does this H2K allele allow for enhanced survival in response to lethal Salmonella challenge? So they took the congenic mice and challenged them intravenously with the Salmonella typhimurium SL1344, which was that control virulent Salmonella, the non-attenuated one. And they analyzed the bacterial burdens after 4 days and then monitored for overall survival. And you can see here that at 4 days post-infection, the H2K congenic mice did not control the initial bacterial growth any better than the H2B congenic controls. They are they're significantly similar. But in the overall survival, you can see that the H2K mice survived significantly longer than the H2B mice. The H2B mice started dying off between 5 and 6 days, and the H2K mice all died at day 8. So therefore, the mice with the congenic H2K allele were able to tolerate infection slightly longer than the H2B mice. So discussion, the results of analysis of the bacteria in this study support the conclusions of the previous studies, suggesting that differences in MHC haplotypes drastically affect primary clearance of Salmonella. CD8 T cells do make a large contribution to protection in H2K allele mice. The depletion of CD8 T cells alone or in conjunction with CD4 T cells had an exaggerated impact on bacterial burdens in mice displaying accelerated Salmonella clearance. This is surprising since the impact of these T cells is relatively modest in the control mice. The protection mediated by the CD8 T cell response implies that different or unique Salmonella antigens are displayed on MHC class 1 molecules, resulting in an amplification of CD8 dependent protective immunity. Since CD8 T cells displayed enhanced interferon gamma production in H2K mouse strains, this cytokine may be responsible for some of the accelerated bacterial clearance observed in these mice. And the study confirmed previous observations that MHC congenic mice displayed differences in the speed of bacterial clearance. However, the study shows that the differences are less dependent on CD4 responses and instead are consistent with the idea of a larger role for CD8 T cells in MHC congenic inbred mice strains. Therefore, there may be a stronger role for cytotoxic T cell activity in clearing Salmonella infections.
Okay, Emily, I'm going to stop you there just because of time again.
Okay.
Yeah, it seems like a really thorough experiment, and obviously it used a lot of immunology to come up with those kind of conclusions. Does anyone have any questions for Emily? So what did you gain from this this type of paper or experiment? I mean, did it get you to understand more about the immune response and dealing with bacteria? What was the most surprising finding of this?
I was really interested in just initially they were talking about CD4 T cells and why they're really important, and that was a lot of the introduction and background for this. And so I think I was just as like surprised as I think they were to find that there was quite a role in the CD8 T cells, and even though it didn't have like a large overall effect, that there were some simple allele changes that could make it so a host can clear out a salmonella infection much quicker. And then also looking at that pro-inflammatory effects that may also aid in the clearance. So I thought it was really interesting seeing how there seemed to be multiple immune responses that were all kind of helping each other to clear out the salmonella infection. And since I work with a lot of salmonella, but in poultry, I think it might be really interesting to see how maybe some of that would cross over.
Yeah, because you want to put bacteria in the MHC class 2 sort of category, but you don't really think about bacteria in the MHC class 1 presentation category. So it shows you that don't look at all sides.
Right, and that was something they had mentioned too.
What's that?
I would say that's something they had mentioned too, was that that was surprising. All right, I just go back here. Oh, it's up here. It's been a while. Stop share.
Thank you. All right, Vanessa, you want to try to give it a try? Oh, there you go. Yeah, you got it. Great. Thanks, Emily. I think that was really interesting. I mean, I think that was well done experiments.
Thank you.
Okay, Vanessa, you're going to be talking about COVID. Great. Yes, we can see it.
I'll try to be quick. So today I'll be presenting a review paper published by Murad and Maytain in June of this year, entitled Pathological Inflammation in Patients with COVID-19, a Key Role for Monocytes and Macrophages. Why it's not passing? Okay.
You just have to oh, you don't.
Okay. A quick overview of what we're going to be talking about today. I will start with a background and then introduction. I will state the objective of the study or the review paper, and then we'll be discussing the COVID-19 immunopathology, followed by the future directions and conclusions. All right, so a quick background for those that did not heard enough. In December of 2019, a novel virus, later identified as SARS-CoV-2, was first reported in Wuhan, China. The disease caused by SARS-CoV-2 then spread across China and across the world really rapidly, and it was declared a pandemic by the World Health Organization beginning of March 2020. In June of this year, Murad and Maytain published the paper that we are going to be discussing today about the pathological inflammation in patients with severe COVID-19. As we saw with Michelle's presentation, SARS-CoV-2 is an enveloped RNA beta coronavirus that contains protein spikes on their surface that allow this virus to bind in the surface of the cell and invade the cell. Humans, bats, cats, dogs, hamsters, they all have been reported to be susceptible to infection by COVID. more than 47 million cases were reported worldwide, with more than 230,000 deaths in the US, more than 1,000 deaths in Kansas, with 12 deaths in Riley County. And as we know, the degree of this disease in infected individuals varies greatly from one individual to another, where the hyperinflammatory response induced by SARS-CoV-2 has been indicated to be the major cause of the disease severity and death in infected patients. So the objective of this paper was to describe the potentially pathological roles of monocyte-derived macrophages during the SARS-CoV-2 infection and applications of this understanding as potential therapeutic strategies. Almost all patients with COVID-19 experience some lung dysfunction, whereas severe complications such as acute respiratory distress syndrome are only observed in subgroups of patients that develop the severe form of COVID. So what makes it so severe in some individuals? They noticed that an increase in blood inflammatory markers, an increase in neutrophil lymphocyte ratio, and an increase in serum levels of several inflammatory cytokines and chemokines are associated with severity of this disease. So what they saw was that the systemic cytokine profile of these patients with severe COVID-19, it is similar of that observed in cytokine release syndrome, which they then called a cytokine storm of COVID-19. Compared with healthy individuals, moderate COVID-19 cases will show an increase in IL-6, IL-10, and TNF alpha, whereas the severe cases will have a further increase in the production of those same cytokines, and plus also soluble IL-2 receptor, TNF as IL-10, and IL-7. In both cases, this will also be accompanied by a decrease in T lymphocytes, particularly CD8 T cells. So their hypothesis then was that a dysregulated activation of the mononuclear phagocytes compartment would contribute to the COVID-19 associated hyperinflammation. Additionally, it has been observed, as I said, that many patients with COVID-19 have a global T cell lymphopenia, which is more pronounced on the CD8 T cells. So not good, since those are our main population that will be fighting viral infections, right? But then the question is this lymphopenia being a result of the direct viral infection of these cells? Would that be a result of a massive recruitment of these cells to the inflamed tissue? Or is that a result of steroid treatments that those patients would be most likely under? They did not know the answer for any of these questions and indicated that it's still poorly understood. Here on the side, it's an image of, not from this paper, but I thought it was cool to share and try to illustrate what I'm talking about, where it shows the degrees of the disease in patients infected with COVID, where you can see CD4 and CD8 somewhat normal in asymptomatic patients. And then you see a lymphopenia in moderate ill patients associated with an increase in IL-6, TNF alpha, and IL-10. And then the storm of cytokine in severe patients associated with a severe lymphopenia of CD4 and CD8, but somewhat more pronounced on CD8 T cells. So they also observed that this T cell depletion was not only in circulation, but also in secondary lymphoid organs. They cited the work performed by Guiding and others in 2020, where they performed a tonal staining, which revealed the high levels of lymphocyte apoptosis in the spleen and lymph nodes of patients who had died from COVID. And that's what it's showing on figure A. This was also associated with an increase of expression of DAF receptor FAS. That's what we see on figure D, suggesting that the activation-induced cell death may be what is causing the lymphopenia observed in those patients. Figure B is just like this, showing the statistical analysis of the figure A, and same as figure E. So they also evaluate the bronchoalveolar fluid from those patients with severe COVID-19. And they observed that was enriched with CCL2 and CCL7. Those are potent chemokines at recruiting CCR2 monocytes. And as I did some research, and apparently those are important for the extravasation and transmigration of monocytes during inflammation. They also observed the presence of macrophage subsets that was enriched with a tissue repair and fibrosis generation genes, like those observed in cirrhosis, and the local clonal expansion of CD8 T cells. In the peripheral blood, they also observed abundance of CD14 and CD16 monocytes producing IL-6. Those are considered intermediate monocytes. Dr. Adams, correct me if I'm wrong on that. And although viral particles of SARS-CoV-2 were detected in mononuclear phagocytes, the evidence of productive infection remained to be demonstrated. In other words, they were still wondering or not sure if the SARS-CoV-2 particles found in those phagocytes were from the uptake of infected cells or from a direct infection by the virus. So they also saw that poor prognosis of patients with COVID-19 was also associated with a decrease in platelets count, an increased level of fibrin degradation products, and presence of other coagulation abnormalities. This inflammation-induced coagulation, it's almost always mediated by expression of a tissue factor or CD142 pathway. And CD142 is expressed on mononuclear cells in response to pro-inflammatory cytokines, mainly IL-6. Also, the oxidative stress was associated with production of oxidized phospholipids, which also induce CD142 expression in inflammatory programmings in monocytes, and activate endothelial cells to recruit and bind to monocytes. So in summary, it seems like it is overwhelming, just like this slide, where the association of activated T cells, increasing expression of tissue factor or CD142, production of those oxidative phospholipids, the increase in expression of inflammatory genes, CCR2, production of CCL2 and 7, and the delayed type 1 interferon response, and plus this cytokine storm. All of that will together lead to this hyperactivation of the monocytes derived macrophages. So as Dr. Adams mentioned before, the balance is really critical for an effective immune response. SARS-CoV-2 has this unique feature of inducing a cytokine storm in infected hosts that leads to a consequent hyperactivation of monocyte-derived macrophages. And it illustrates how the pro versus the anti-inflammatory activation determines the protective versus the deleterious impact of this response on the host. So as a future direction, this characterization of the immune and inflammatory response in patients with COVID-19 is critical for the development of effective diagnostic methods, therapies, and vaccines against COVID-19. They say that clinical trials assess the of inflammatory cytokines targeting IL-6 and IL-1 alone or in combination were described to be already in process, among other possible therapeutic targets associated with activation, such as the anti-tumor necrosis factor, CCR2 antagonist, and anti-interferon gamma.
Yeah, it looks like there's lots of different options as far as, it seems like they've honed in on a lot of important mechanisms that they could potentially use as a target for therapy.
Yeah, definitely.
Yeah. Does anyone have what's that?
So sorry, I thought that illustration is really overwhelming, but really nice to see the possible targets that you can develop on therapeutic treatments to block those pathways.
Yeah, I think this is the first that I've heard about, looking really at the immune response to what's going on with SARS-2. Does anyone have any questions for Vanessa? It would be interesting to see, what therapeutics have worked, because like you said, it's this balance sort of situation. You don't want the body to not fight off the infection, but you also don't want it to go berserk at the same time and cause the cytokine storm. I guess I didn't realize that there were two layers of cytokine responses, the primary, and then there's a secondary that's kind of overwhelming the system, sort of thing.
Yeah.
And it's also interesting that they were looking at CD8s that were sort of disappearing, and that they were suspecting it was probably due to a massive amount of apoptosis.
Yeah, that was actually one of my quiz questions, that where they attribute that overproduction of IL-6 to the decrease or the suppress the T cell activation, which I believe them. I'm not sure, but that's what they said, that IL-6 would be a potent when overproduced, it would suppress the normal T cell activation.
T cell activation. So that would include the CD8 cells.
Right.
I see.
That's what I understand, I think.
Yeah, it's quite a it's quite complicated as you mentioned and as you illustrated. But yeah, I think that was
like a nice.
that definitely
they had a nice table at the end.
Yeah, I mean, I think the diagram helps it help explain just how complicated the immune system can be, and especially for diseases that are unknown. We're sort of starting brand new and not, we don't know everything that's happening. So it's just trying to figure out what the model is. So then you can really think about targeting some therapeutics against the key triggers for that, um, huge immune inflammatory response to the viral infection. So yeah, it's pretty interesting. Any questions for Vanessa? I think we better wind it down. I think I'm running out of time already. So yeah, these were really, really interesting and great presentations. So thanks again for our speakers. I will be sure to post those slides in the last module so that you can have access. And obviously you'll see that there's some folks that have put in their quizzes at the end. So be sure to have a look at that. And I guess with that, I guess we'll stop for tonight. I did get some things graded today, so be sure to have a look at those. And if you have questions, let me know. Otherwise, I think we better stop for tonight, and we'll see you next week. Next week, we'll have Dr. Mwangi talking about tolerance, autoimmunity, and hypersensitivities. So that will play well along with what Shelley was presenting today. And then we'll have another set of presentations next Wednesday. And then there's also an online quiz on lectures 14 and 15. So, but we'll worry about that next week and let you know about that more. With that, I think we better stop. Hope you guys have a great rest of the week. And hope you can rest up after the big election night last night. So, so hope everyone has a great night. Thanks, everybody. Thanks, presenters.
Thank you.
Thank you.
All right. Bye.
Bye.
Bye-bye.